My name is Tasuku Honjo. I'm a distinguished professor in Advanced Institute in Kyoto University. Well, cancer immunotherapy started only 2014, the first time approved the drug that blocks the immune system called PD-1. We developed the antibody against this immune brake, and that activate immune system, and now the immune system can recognize the tumor as not self and try to eliminate. And we are now working on this line. Still, this is just beginning. There are several problems. One, it's not effective to all the patients. Currently, 20–30% of patients responding in case of lung cancer. There are variations among different cancers. And also, because it's not 100% effective, we wish to identify or find the mechanism why each individual patient have different response. That's what we're doing now. So all organisms have some kind of immunity against the pathogens. There are two different types of immunity. One is called innate immunity or natural immunity, and the other called acquired immunity. Innate immunity is evolutionary earlier. All organisms have this mechanism. The big difference between innate immunity and acquired immunity is twofold. Acquired immunity is evolved very recently. Only vertebrates have this system, and it recognizes very fine difference of pathogens or other molecules which invaded our body. But innate immunity recognizes difference of the so-called pattern recognition. It's not a precise difference. So in that sense, acquired immunity has very specific and accurate recognition. And that's why it's useful to recognize tumors, because tumors originate our own cell but accumulate mutations that generate something which didn't exist in our body. So it's a subtle difference. Yes, microbiota in our gut has a very mysterious, still not fully understood, but important function. First of all, the microbiota is under the control of our immune system. Immune system secrete IgA antibody into the gut lumen, and that binds bacteria and keep so-called beneficial bacteria and try to eliminate unwanted bacteria. If this balance is disturbed and unwanted bacteria expand, somehow immune system sense, and then the whole body immune system get activated. And that further leads to the very dynamic interaction with other regulatory systems, including nervous system, whole metabolite regulation. So this field is just beginning, and we have to work, keep working. But certainly it's a very hot issue, And when we work on immune system, dependency or mutual regulatory system between microbiota and immune system is very important. That's a very good question, and actually I'm very optimistic. When penicillin was discovered in the infectious field, it was certainly dramatic, but it's not cured all the infectious diseases. Subsequent, I don't know, maybe half century, series of new generation antibiotics discovered, and now we think we can almost all the infectious diseases are under the control. So cancer immunotherapy is just beginning, only 20%, 30% lung cancer cured. Currently, 1,000 clinical trials are on the way to find to better improved immunotherapy. Most of the people, I mean the scientists and the company, try to add something to cancer immunotherapy and improve the efficacy of the treatment. So given this condition, I expect in 10 years, the percentage of the responders will increase, and hopefully, I don't know how many years, eventually we hope cancer may not be such a life-threatening diseases. In some cases, we don't have to completely abolish tumor. If we can control the growth of tumor, we can live with the tumor, especially those elderly people. They don't have to fight. They can live with cancer under the control. That's what I hope. I don't know it. Hopefully within this century. This is the first postdoc I took, and Carnegie paid my study here and offered very nice environment, and especially the interaction with Don Brown had a very strong impact on me. He told me the long-term question of how to generate immune diversity. Now this huge question can be the actual target of the scientific approach. He explained DNA studies going on, reverse transcriptase may allow us to generate cDNA and eventually DNA cloning, and we are just lucky, hit the right moment to attack one of the biggest questions in biology, and that led me to work on the immunoglobulin gene. And fortunately, Phil Leader, who were at that time NIH, came to Carnegie and gave a talk, and I immediately met him and asked him to move to his lab. So that's the beginning of my whole career. So I have still scientific questions. One is the molecular mechanism of AID, which I found 2000 as the enzyme that regulate class switch recombination and somatic hypermutation. And second is the PD-1, which I'm working on to improve the efficacy of anti-PD-1 treatment, and also to find the markers to distinguish responder and non-responder. Outside the science, I wish to set up the fund to support young people. I already made a fund in Kyoto University, and we began to obtain several donations, but the biggest donation I expect is from the pharmaceutical industry, who use our patent about PD-1 treatment. So if the endowment become big enough, I like to use this money to support young scientists. So no obligation for 5 years, and they can hire several postdoc and cover the expenses. That's my dream.